Can anyone hear me? I didn't
I
hear
can
anyone
hear
come
you.
on. Let's see if my microphone is on.
I can hear you.
No, my speaker's off. All right. There, can anyone hear me now?
Yes.
Yes.
Thank you. Uh, it's interesting. The background's now working when I have the light down dim and I don't even have the green screen behind me. So that's interesting. Anyways, before we begin, are there any questions? All right, let's begin. Share my screen here. Guess we can close that down.
Actually, I had a question about the test tomorrow,
Uh-huh.
and I just wanted to verify the sections of chapter 2 it's going to cover. It's not going to cover the first section, correct? I thought that you had said that.
Quiz 1 covered chapter 1 and 10 and the start of chapter 2. Then lab 0, 1, lab 0, lab 1, and lab 2. Quiz 2 is going to cover labs 3 and 4 and chapters 3 and the end of chapter 2. So if you want to know what's on the quizzes, I always have it up here in the syllabus, what the quiz will cover.
Perfect. Okay, I just wanted to make sure I was following that so I could
All
be
right,
prepared.
and let me state that I just got, I think it was lab 4 graded. So I did get all of the last week's lab. That can't be right. Just a minute here. I can't be right because it can't be lab 3 and 4. So maybe it's lab 5. Let me take a look at the grading here. I'm going to shut down the screen because I'm going to open up the grading book. Come on. There we go. I thought I got both of last week's labs graded, and I just finished right before class began. I just finished the last one. Yeah, lab 4 and lab 5 from last week are now graded for the first time. The class has voted to have the labs graded 2 times. Let me make sure I've got that in there. Yeah, you have until this Saturday to grade lab 4 for the second time, and then lab 5 as well. Oop, that's not right. Let me change that date there. It's got August 9th, and I don't think it's that long. That would be over a week, so I think it's August 2nd. Yep. Most of you missed lab 5. Not sure why that's the case. Meaning very few people got 100. I don't think anyone actually got 100 on lab 5, but I could be mistaken. So take a look at that. You do have a second grading. Some of the questions are thinking problems, and you have to come up with a thoughtful answer. Some of you are sharing your answers with other people, and if you don't share the correct answer, then both of you will get it wrong. All right, actually on lab 4 and 5, nobody got 100%. That is rare. Usually I get students who get 100%. All right, any questions about any of that?
Um,
Yeah.
So for lab 5,
Yeah.
oh, it says I got 5 out of 5, but it says I got one question wrong. So I'm confused.
Did I make a comment saying that you can miss one question on the first pass of the lab?
Yeah.
All right, that explains it. All
Okay.
right,
Thank you.
let's move
Sorry.
on. Yeah, let's move on. Um, I thought I just looked at the lab 5 and I didn't see 100 and 100, but maybe I missed one. Let's come back to the lesson. So what we're going to do today is July 29th, and August is almost about to begin. Um, we're supposed to be finishing up chapter 4 and beginning chapter 5. There is a lab today. Um, and that's it for what we're doing today. All right, let's move on to chapter 5. I have that we had discussed the cytoplasm last time. Cytoplasm being the liquid inside the cell membrane. It's mostly water, but it does have many dissolved substances, as well as some things like ribosomes, which are too big to dissolve in the cytoplasm. The nuclear area, also called the nucleoid region of a prokaryote cell, is wherever the single chromosome of DNA is located. It floats around in the cytoplasm, and wherever it is called the nucleoid region. It does give the cell different staining properties, but other than that, there is no distinction between the nucleoid region and the cytoplasm because there's no barrier between the two. It's wherever the DNA happens to float in the cytoplasm. You can also have plasmids in the cytoplasm. That's a small molecule of DNA, and we'll talk about that later in the term. Ribosomes are large molecules made up of protein and rRNA, and you could call them ribosomal protein and then ribosomal RNA, make the ribosome. They are so large they cannot go in solution in the cytoplasm. The ribosomes of a prokaryote are a little smaller than the ribosomes of a eukaryote. The ribosomes of bacteria and archaea are 70s in size, and the ribosomes of a eukaryote cell are 80s in size. The S is just the sedimentation coefficient, where the ribosome sediments in the ultracentrifuge gradient. And that's just the way biochemists and I guess people who study ribosomes differentiate ribosomes. Any question about any of that? The ribosomes are where the cell makes its protein, so you can call them the protein factory. Ribosomes are made of two parts. You don't need to know the different parts. You should know that they are made of protein, ribosomal protein, and ribosomal RNA called rRNA. There are also inclusions in the cytoplasm. These are reserve deposits within the cell. They're stored in the cytoplasm. And when enough of this material builds up at one place, there's so much of it that it precipitates out of solution. Cells tend to use these resources when resources are low, and usually cells store a material that the cell can metabolize. There's different examples of inclusions. You don't need to know the different names of the examples. Oops. Here we're looking at some of the different inclusions. Magnetosomes here. Let me blow that up so you can see it better. Little dots going like that. This cell metabolizes sulfur, so it's storing sulfur inside it, so much sulfur that it precipitates out of the cytoplasm. And you can see the cells are almost full of sulfur. And then metachromatic granules, you can sort of see the yellow line around the cell there. And there are the inclusions, 3 of them in each cell. Well, at least these 2. All right, any question about inclusions? They're just material accumulating in the cell, that so much of it is in the cell that it actually precipitates out of solution. And then you have this solid mass in the cytoplasm that we call an inclusion. The next topic I want to talk about are endospores. This is the second time we've talked about endospores. We will talk about them 3 times in the class, so obviously this is an important topic. Endospores are structures a cell makes to survive. So they are survival structures. They are not reproductive structures. Usually one cell makes one endospore, and then the parent cell dies. So one cell will then result in one endospore, and that's not reproduction. They are a specialized resting cell produced by certain species of bacteria, mainly Bacillus and Clostridium, those 2 families. They allow the cell to survive in harsh environmental conditions, such as desiccation, heat, chemicals, radiation, and they can survive conditions that the vegetative or parent cell cannot. So when the environment becomes deteriorating, the parent cell will notice the deteriorating environment and say, hey, I'm going to be dying pretty soon. And so it'll then start making an endospore. And there we see an endospore inside a parent cell right there. Sporulation, also called sporogenesis, is when the cell is making an endospore, meaning endospore formation. And then when the environment becomes favorable, the endospore can germinate and return to the vegetative state, meaning a vegetative cell, which is similar to the parent cell, and it can grow and reproduce. Endospores have very little metabolism going on in them, very little or none. And that's one reason why they can survive. They also have less water than most living cells. Not sure if that helps them survive or not, but they do have less water too. Excuse me here. Wanted to blow my nose. All right, any question about endospores? Endospores form by the cell dividing in 2. When you're making an endospore, the cell doesn't divide down the middle. It will begin to divide with a smaller cell and then a larger cell. The smaller cell then gets a plasma membrane surrounding the DNA. The DNA, of course, divides first and goes to both sides of the cell. And then you get 2 plasma membranes around the structure that will become the endospore. You note the endospore is now moved in a little ways. That sometimes happens in endospores. And then the endospore forms a thick peptidoglycan layer between the cell membranes. You then put a spore coat on the endospore, surrounding with the thick spore coat. And then the parent cell dies, which, if you notice here, the cell was dividing into 2. This part becomes the parent cell, which then gives rise to forming the endospore. Parent cell then dies, and that will release the endospore, which can survive harsh environmental conditions. Here we have a little movie of an endospore forming It's not that good, but it does show you an endospore forming. If it starts, there we go. This cell is going to form an endospore. Oops. Let's back up a little ways. There, it's starting to form an endospore. All right. Any question about any of that? Remember, an endospore is not a reproductive structure. It is a survival structure. All right, that's it for the structures of prokaryotes. Let's talk just briefly about, oops, that's not going to work. Let me blow it up this way. The structures of a eukaryote cell. We do have differences between a plant cell and an animal cell. The animal cell doesn't have a cell wall around it, just a cell membrane. It tends to be more circular than a plant cell. The plant cell has the shape of the cell wall. In this one, it looks like the cell would be hexagon. You do have different structures in plant cells and animal cells, but most of the structures, especially the membrane-bound organelles, will be found in both plant and animal cells. Like both the animal cell and the plant cell can have a flagellum. They both have cytoplasm. They both have a nucleus. They both have Golgi apparatus. They both have mitochondria. Only the green plant cells have chloroplasts. No animal cell has a chloroplast. Any question about any of that? Now I said a green photosynthetic cell of a plant has chloroplasts. Does that mean there are cells of a plant that do not have chloroplasts? It's not a trick question, people. Does the root cells of a plant have a chloroplast?
No?
Yeah, that's correct. The root cells don't normally see sunlight, so they don't have chloroplast because they don't engage in photosynthesis. There are some organelles or membrane-bound organelles that differ between the plant cell and the animal cell. Like a plant cell has a central vacuole, that's that one there, and it's labeled as a vacuole. That's not quite correct because both animal cells and a plant cell have a vacuole, but only the plant cell has a central vacuole. The central vacuole is the largest organelle in a plant cell, even larger than the nucleus. I don't know if that's actually showing here, but an animal cell, the largest organelle is the nucleus. By organelle, I mean membrane-bound organelle. Any questions about any of that?
No.
Okay. There's an non-membrane bound organelle. I don't like that term, so I call it a cell structure that is found only in animal cells. The centrosome containing the centriole in it, and this in animal cells tends to make the mitotic spindle when the cell is about to divide, either in mitosis or in meiosis. It's curious because a plant cell does have a mitotic spindle, but it does not have a centrosome to make it. All right, so those are some membrane-bound organelles as well as non-membrane-bound organelles that differ between plant and animal cells. And there we're looking at an actual electron microscopic image of a plant cell on this side. and an animal cell on that side. Any question about any of that? All right, I've got the different structures. Let me shrink that down a little bit. Oops, that's not right. I wanted to do. Of a eukaryote cell in the slide. You can go through it if you wish. If you remember it perfectly from your preliminary class before taking microbiology, that's fine. I'm not going to go spend the time on this. I do want to talk, though, about one thing. Well, actually, two things. Let's go down here. Eukaryote cells have an endomembrane system where various membrane-bound organelles are part of a cellular transport system, being a conveyor belt for cellular molecules moving around in the cell. Especially proteins are moved around in the cell, and proteins usually start in the rough ER, and then a transport vesicle takes the protein to the Golgi apparatus, which modifies the protein, and then a transport vesicle moves the molecule.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the
then moves the molecule to another part of the cell. And while the transport vesicle is moving, it's called a transport vesicle. But if the transport vesicle then starts storing the molecule and then stops moving, we don't call it a transport vesicle anymore. It's called a vacuole. Any questions about any of that? So the only difference between a transport vesicle and a vacuole is a transport vesicle is actively moving a molecule around or several molecules around in the cell, and a vacuole is not moving, it is just storing the molecule. All right, let's watch the endomembrane system.
endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the trans face of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Moo Math uploads a new math and science video every day. Please subscribe.
Okay, any questions about that? And the transport vesicles coming from the ER actually bud out of the ER. If they're carrying proteins, they come from the rough ER. If they're carrying lipids, they come from the smooth ER. And then that vesicle buds or binds to the Golgi apparatus. And then the Golgi apparatus modifies the molecule, either the protein or the lipid. And then the a vesicle buds out of the Golgi apparatus, the other end of the Golgi apparatus, and will carry the molecule to where the molecule needs to go in the cell. Remember, the vesicle is a transport vesicle if it's carrying something and moving in the cell. If the vesicle stops moving, we then call it a vacuole, and it's just going to store the molecule in the cell. Any question about the endomembrane system?
I don't have any.
There's a picture of the endomembrane system. Okay, let's move on. Let's talk about the endosymbiotic theory. This is a theory to explain the evolution of mitochondria and chloroplasts in eukaryote cell. These organelles differ from other organelles because they have their own DNA molecule in the membrane-bound organelle. They also have their own ribosomes within the membrane-bound organelle. And they have a double membrane, meaning they have both an inner and an outer membrane. Any questions about any of that? How did they originate? Well, they think a long time ago, we had a proto-eukaryote cell. It was very big. And it was a eukaryote cell before we had eukaryotes. So that's why I said a proto-eukaryote cell. And it had no mitochondria or chloroplasts. It also engaged in fermentation. Let me spell that out. To get its ATP. Meaning it took one glucose and used fermentation to get 2 ATP. We'll hit another little cell over here. Well, let's begin this one right here first. And I'll draw the little cell in a minute. But this cell used aerobic respiration to get its ATP. And so it took one glucose and made about 38 ATP from one glucose. And this was a little cell like this. And erase that now, move this closer. One day the big cell came upon the little cell and then engulfed it. So let's make the little cell over here. And how it engulfed it was pseudopods grew out here and here. And then the pseudopods grew together and then fused. I should draw this where they're growing together. I guess I can do that. And then I'll fuse them together. Oops, let's move that over here a little more. And there should be two there, but I'm just going to draw one. And then they fuse together. And now you can see that the little cell is within the big cell. And furthermore, there's two membranes around the little cell. It's its own membrane right here. And then there's the vesicle membrane of the proto-eukaryote cell. And let's go. Oops, that's not what I wanted. And this over here was actually a bacteria. For whatever reason, the proto-eukaryote did not immediately engulf or eat the bacteria cell that it engulfed. It kept it around for some reason and probably was just leaving it as a reserve food supply and was probably intending to use it later. Now, this obviously can move deeper in the cytoplasm. Let me move that with this here. And probably because life was young at the time, some of the glucose in the cytoplasm went from the proto-eukaryote to the bacteria cell and just diffused around. And then the engulfed cell would move it in across the membrane with a transport vesicle. And the bacteria cell, probably because life was young, shared some of its bacteria with the proto-eukaryote. It might not have done it in purpose, it's just that there was liquid around and the glucose moved from the engulfed cell to the proto-eukaryote, meaning from the bacteria to the proto-eukaryote. And the proto-eukaryote noticed it lost one glucose molecule, but instead of gaining 2 ATP, it gained a number of ATP. We don't know how many ATP this cell initially shared with the proto-eukaryote, but it was something much greater than 2. It probably was a number less than 38, but much greater than 2. And the proto-eukaryote was confused. It said, I lost one glucose, but I gained much more than 2 ATP. And it came to realize that this bacteria that it engulfed was making ATP for the proto-eukaryote. And so it decided, well, I'm going to keep this little cell around. I'll feed it glucose and then it'll make me ATP, much more ATP than I can make. And then the two started living in, what's the term? Symbiosis, living together. They've now lived so long together that we find almost all proto-eukaryotes have a cell inside them that makes ATP. And then the cell inside them, we don't see free living anymore in the environment. The cell inside we only find within a proto-eukaryote. And since the two have lived along for so long together, We're not going to call it a proto-eukaryote now. We're going to call it a eukaryote, because it has a cell inside of it making ATP for the eukaryote cell. And that is how they think the mitochondria and the chloroplast came about in a eukaryote cell. Any question about any of that? What proof do they have that this story came about? Well, first, the mitochondria and the chloroplast have two membranes, the same as a vesicle containing a cell, meaning you have the vesicle membrane and the cell membrane of this cell here. So that's one piece of evidence. The other evidence is that there's DNA inside this bacteria cell, and there's DNA inside the mitochondria and the chloroplast. Let me see if I can remember what some of the other pieces of evidence are. Oh, yeah. Second, there are some eukaryotes, simple eukaryotes, that actually engulf another cell and then use that cell to make either ATP or glucose for the protist. It's a simple eukaryote, the protist. And there is some amoeba which lack a mitochondria. And these amoeba engulf a bacteria and then use that bacteria to make ATP. So these amoeba lack mitochondria, which makes most of the ATP in the cell. And then these amoeba engulf a bacteria, which makes ATP by aerobic respiration, and then share some of that ATP with the amoeba. So we actually can see this, I guess, symbiosis occurring between the early eukaryote cell and then another cell. Here we have a bacteria which is engaging in photosynthesis and a protist, and the protist will give light to the photosynthetic bacteria as well as nutrients and CO2. And then that photosynthetic bacteria will engage in photosynthesis and share some of the glucose with the protist. So we actually do see this happening in life today. And then some of the other evidence, the DNA of a prokaryote cell tends to be one circular molecule, at least to a bacteria. And in a eukaryote cell, all of the chromosomes are linear. But in the mitochondria and the chloroplast, the DNA is a circular molecule, same as the bacteria. And then there's no histones on the DNA of a bacteria cell. There are histones on the DNA of a eukaryote cell. There are no histone proteins on the DNA of a mitochondria and the chloroplast. If we look at the first amino acids synthesized in a eukaryote cell, it's methionine. AUG stands for the amino acid methionine. In a bacteria, it's formylmethionine, a slightly different amino acid. And then we look at the first codon of a mitochondria and a chloroplast, it's formylmethionine. When we look at the ribosomes of a eukaryote cell, they're 80S. The ribosomes of a bacteria are 70S. When we look at the ribosomes that are found in a mitochondria and the chloroplast, they are 70S. And then the prokaryote cell mainly divides by binary fission. The mitochondria and the chloroplast divide by binary fission. The eukaryote cell divides by another mechanism that we call mitosis. And then the strongest evidence for the mitochondria and the chloroplast being bacteria is when we sequence the DNA of a bacteria, the DNA falls within the domain bacteria. When we sequence the DNA of a eukaryote cell, the DNA follows the sequence of a eukaryote. When we sequence the DNA of a mitochondria in the chloroplast, they follow the sequence of a bacteria. Any question about anything? All right. What's a little unusual is that this evolution in plants actually happened twice. First, the plant as well as the animal cell engulfed a cell which made ATP and became the mitochondria of the eukaryote cell, both the plant and the animal cell. And then the plant engulfed another bacteria, which after symbiosis and living together for many years became the green plant cell's photosynthetic chloroplast. Any question about any of that? All right. Anyways, that's the theory of why eukaryotes have mitochondria and chloroplasts. There was an early prokaryote cell, an early eukaryote cell, which engulfed the bacteria. One became the mitochondria, another became the chloroplast. Any question about any of that? All right, let's move on to the next chapter then. Chapter 5. Which we are supposed to start today. All right, the first slide shows you the major goals and a rough outline. If you want to know all of the goals, you should look at the objectives, which are in my notes. Oop. Study guide. Yep, I didn't have chapter 3 slide published. If you don't have it published within a day of finishing it, let me know and remind me and I'll turn it on. Let's take a look at the study guide. The very first part of that will have the chapter objectives. Right there. Let's close that down. Know the terms. What's catabolism, anabolism, activation energy? What's an enzyme? What's an active site on the enzyme? What is competitive inhibition, non-competitive or allosteric inhibition? What is a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotroph, and chemoheterotrophs? What is fermentation? And what is cyclic and non-cyclic photosynthesis? Understand what feedback inhibition is. And 3, understand what aerobic respiration is, know its steps, and how it differs from anaerobic respiration. Any question about what we're going to cover? All right. When we are talking about the metabolism of an organism or a cell, we're talking about all of the chemical reactions within the cell. And when we're talking about microbes, the cell and the organism are the same. You can separate metabolism, all of the chemical reactions within the cell, into two large groups, catabolism and anabolism. Catabolism is the decomposition reactions, starting with a larger molecule, such as starch, and then breaking it down into a simpler molecule, such as glucose. Energy will be given off by this reaction because there's more energy in the complex molecule than in the simple molecule. Some of that energy given off is always converted into heat, and that's one of the laws of thermodynamics. Whenever energy is transferred from one form to another, some of that energy will be converted into heat. Some of that energy can be given from the complex molecule to the smaller molecule, can be used to make ATP. You make ATP by phosphorylating, meaning you add a phosphate to ADP to make ATP. Now understand that animal cells are lousy at making ATP. We only have two mechanisms for making ATP. I guess I'm talking about humans, but generally all animal cells, and that is aerobic respiration and then fermentation. The only two ways that we can make ATP. Even green plants, which have more ways of making ATP, are pretty lousy at making ATP. Only a few of the catabolic reactions in a green plant can be used to make ATP. Most of the catabolic reaction, and in animals, all but two of the catabolic reactions do not generate ATP. So this is a little oversimplified because in animals, only two of the catabolic reaction, meaning aerobic respiration and fermentation, can be used to make ATP. All the other catabolic reactions cannot be used to make ATP. An example of a catabolic reaction that cannot make ATP is the splitting of sucrose into glucose and fructose. That is a catabolic reaction. Energy is given off by the reaction. You have a larger molecule being split into two smaller molecules, but ATP cannot be generated. Any question about any of that? All right. Anabolism is a biosynthetic reaction where you take a simple molecule, such as glucose, and then you make a larger molecule, such as starch. You need energy to come into the system to make this reaction occur because starch has more energy in it than molecules of glucose. So energy is required for this anabolic reaction to occur, and that energy usually comes from breaking down ATP, releasing the energy needed to have this anabolic reaction occur, and then ATP is broken down into ADP and phosphate. The I stands for inorganic phosphate. So energy is required to convert glucose into a larger molecule such as starch, but some of that energy will be given off as heat because the second law of thermodynamics states that whenever you convert energy from one form or another, meaning here ATP and glucose, into the energy in starch, some of that energy is always converted into heat. The exact term is energy is lost as heat, and energy is not ever lost because heat is just another form of energy. Any question about any of that? All right. Let's talk a little bit about metabolic pathways. We're looking at a metabolic pathway here. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell. So one enzyme can convert the molecule A, which is the starting substrate, into the molecule B. Molecule B is the product of this chemical reaction. But then molecule B can be converted by another enzyme into molecule C. In this case, molecule B becomes the starting substrate of this chemical reaction. And then molecule C is the product of this chemical reaction. But molecule B was the product of this reaction, and that's why it's called an intermediate product, because it is an intermediate, because in this reaction here, molecule B will be the end product of this one, but the starting of this one, and molecule B will be converted to molecule C. And then you can have another enzyme converting molecule C to molecule D, a fourth enzyme converting molecule D to molecule E. And all of these molecules here are called intermediate products because it's an intermediate molecule between this one and that one. And this molecule is a product of this chemical reaction, but the starting substrate of this chemical reaction. Well, they're called intermediate products, or just intermediates. And then molecule F is the end product of the whole metabolic pathway. The last molecule, that molecule A would be converted to this, to that, to that, to that. And this will be the end product, the last part of this. Any question about any of that? All right, metabolic pathways are determined by enzymes, and enzymes are encoded by genes. So the metabolic pathway happens in a cell because of the genes in that cell. Any question about any of that? All right, this is showing you a huge amount of metabolism that we're going to talk about in today's lesson. Any questions? Only kidding, we're not going to talk about the huge amount of metabolism occurring in the cell, and this is showing you the, well, metabolic reactions occurring within a cell. Let's talk a little bit about enzymes and how they work and how the product is converted into, the starting substrate is converted into the product. This all happens by the collision theory of chemical reactions. Chemical reactions may occur when atoms, ions, or molecules collide with one another. And that's the collision theory. So we get chemical reactions occurring when molecules or atoms or ions collide with each other. Activation energy is the energy needed to disrupt the electronic configurations to convert the starting substrate into the product. So if the collision of the two molecules have enough energy, the activation energy will be reached, and then the starting substrate will be converted into the products, or product, depending on the reaction. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes. The more enzymes you have, the enzyme then will bind to the starting substrate more quickly, and then convert the starting substrate more quickly into the product, increasing the reaction rate. You can also increase the reaction rate by increasing the temperature or the pressure. If you increase the temperature, you're making the molecules hotter, meaning the molecules are moving around more. So the hotter the temperature, the more the molecules move. The more the molecules move, then the more likely you're going to get the collision with the correct activation energy to make the product. Any question about any of that? All right, temperature, I think you guys understand quite well. The higher the temperature, the greater the reaction rate, just because the molecules colliding have more energy, so they're more likely to have the energy they need for the activation energy. The same thing with pressure. If you increase the pressure, then the molecules are actually closer together or more pressurized, and the closer they are together, the more likely they're going to collide, and that will increase the reaction rate because you increase the collisions per second, I guess. It also increases the energy that the molecules have, and that's because they're under pressure, and that also increases the reaction rate. Any question about any of that? All righty, this is showing you an enzyme performing a chemical reaction, and we have a reactant, the starting substrate. It has a higher energy level than the products. Like if this is glucose, the glucose molecule has more energy in it than CO2 and water, the products of this reaction. Because the initial reaction is higher in energy than the products, this reaction is going to tend to occur. However, before that reaction can occur, the molecules colliding have to have enough energy to disrupt the bonds, the chemical bonds in this molecule, to make the products. And this we call the activation energy without an enzyme. If AB was, for example, glucose, we could convert all of that glucose into the products, CO2 and water, simply by taking a blowtorch to the glucose and then setting the glucose on fire and burning it. That tells you that you need a lot of energy without an enzyme, meaning the activation energy without an enzyme is very high, much higher than the activation enzyme with the activation energy with an enzyme. One example of this, like I said, to convert glucose into CO2 and water, you can get a blowtorch. That's around 1,000 degrees, either Fahrenheit or Celsius. I forget which, doesn't matter, it's very hot. To get the glucose into the activation energy we need to convert it into CO2 and water. And yet, in the room that you're in, this reaction with glucose to CO2 and water is happening all the time. And where is that happening in the room that you're in? Come on, somebody's got to guess. Where in the room that you're in are glucose being converted into CO2 and water? Well, how about in your cells? Do your cells make CO2 and water from glucose?
Yes.
Yes. Obviously, inside your cells, you do not have an activation energy of 1,000 degrees Fahrenheit, because that would fry your cells. And the reaction activation energy with an enzyme is much less than the activation energy without an enzyme. And the activation energy with an enzyme, at least in your cells, is around 37 degrees Celsius. That's 98.6 degrees Fahrenheit. Much less than 1,000 degrees for taking the blowtorch to the glucose. Any question about any of that? All right. Enzymes are biological catalysts. They speed up chemical reactions, 10 to the 8th to 10 to the 10 times. You don't need to know the numbers. You just need to know that enzymes are biological catalysts and they speed up chemical reactions. Enzymes are generally specific for a single chemical reaction. So most enzymes work on one molecule and one molecule alone. For example, the enzyme for breaking glucose is a different enzyme than the enzyme for breaking either fructose or sucrose. It is true that some drugs and some other enzymes can work on more than just glucose. But for our purposes, we'll oversimplify it and just say that an enzyme, essentially speaking, is specific for one starting substrate. The specificity of the enzyme is due to its three-dimensional structure. Each enzyme acts on a specific substrate with a specific part of the enzyme. And then the enzyme is not used up by the chemical reaction. Once glucose is made into CO2 and water, that enzyme is released, and then it's free to bind to other glucose to carry out its chemical reaction. Any question about any of that? Oops. I should get down just a little. So an enzyme converts the substrate to the product largely by the enzyme forming a complex with the substrate. And when the enzyme forms a complex with the substrate, that puts stress on the chemical bonds of the substrate, converting the chemical bonds of the substrate to another molecule, such as when we're taking sucrose and we're digesting it to glucose and fructose. We have an enzyme to help that chemical reaction. So the enzyme is binding to the substrate pretty specifically. I don't know of any drugs in place at the moment, but I'm sure there's one developed that does work on this chemical reaction, like maybe a drug that they give diabetics. I don't know. The point is the enzyme substrate complex orients the substrate, increasing the probability of a chemical reaction, also lowering the activation energy, and then making the substrate change into the product, largely by changing the chemical bonds are within the substrate. Any question about any of that? All right, enzymes tend to end in ACE. All enzymes that were discovered after this convention came into play, the enzyme ends with ACE. Here we have a few. You don't need to know these. We will talk about ligase. If we haven't already talked about it, have we talked about it? Have we talked about DNA replication? I'm not hearing anything, so I think it's a future lesson. I don't remember. Maybe we've talked about it. And then I've talked about catalase in the lab. These are two enzymes that end in ACE. When we're talking about enzymes, not all enzymes end in ACE, like renin, the enzyme in the stomach, which converts liquid milk into a solid, carries in whey, and the baby has this renin because babies drink a lot of milk, and having the renin in the milk makes the milk thicker, so it's more likely to stay in a patient, so the patient can digest greater amounts of the milk. Any question about any of that? All right, you don't need to know the enzymes that end in ACE, but we will talk about ligase later in this term, and we will talk about catalase as well later in the term. When we're talking about an enzyme, excuse me here, when we're talking about an enzyme, it's important to realize that some enzymes have a helper. The helper is either called a coenzyme or a cofactor. A coenzyme is a biological molecule within the cell that actually binds to the enzyme and forming the active site. You do not need to know the term apoenzyme and holoenzyme, but you should know coenzyme and that it binds to the enzyme to make a complex, which then allows the substrate to come and bind to the active site of the enzyme. Before the coenzyme binds to the enzyme, the substrate cannot bind to the active site of the enzyme. And that's true of a cofactor too. A coenzyme, as I mentioned, is a small biological molecule, like NADH is a coenzyme. A cofactor is a non-protein molecule which binds to the enzyme and then activates the enzyme, usually by changing the three-dimensional shape of the enzyme. Any question about coenzyme and cofactor? NADH is an example of a coenzyme. A cofactor would be something like copper or iron or even magnesium, something that our bodies need in very low amounts. And we usually get this from the environment. One exception in Western Oregon and Western Washington is because of our rain, it leaches out from the soil the nitrogen compounds, and then the nitrogen compounds are washed down to the stream, washed to a river, and then washed out to sea. So an enzyme doesn't want that to happen. And one thing it does is it quickly binds to the starting substrate and then perform its chemical reaction. And it may, if it's a triangular type coenzyme, it may stay with the product until the chemical reaction is done. It may or may not need a structure to bring the starting substrate and the enzyme together. It may not need that. I'm not sure what I was getting at, but hopefully I covered that. This slide is showing you some important coenzymes. You do need to know the first two because we're going to talk about these in aerobic respiration in our next lesson. But NAD plus, or nicotinamide adenine dinucleotide, is a coenzyme that some enzymes require. And then FAD, flavine adenine dinucleotide, is another important coenzyme. we'll talk about these two coenzymes when we're talking about aerobic respiration. The other coenzymes, I've got them listed. You don't really need to know them. I won't quiz you on the other coenzymes, but do realize that like in green photosynthetic plants, we don't have NAD acting as a coenzyme. We have NAD phosphate with 1+ charge being the coenzyme or cofactor. And yes, those two terms can be used interchangeably, although realize that they don't have to have the same meaning. Any questions about any of that? All right, this is showing you the different ways you can represent an enzyme in a enzymatic reaction. You can discuss the enzyme as a key that specifically fits one lock, and this fits specifically one substrate, putting stress on the substrate when the enzyme substrate complex forms, and that stress helps convert the chemical bonds in the substrate into the chemical bonds of the product. And this is known as the lock and key model of how an enzyme functions. There are different models, and this one here is known as the, what is this one called? Let me think about that. I just drew a blank. I had it on the tip of my tongue and then I forgot. I have to think about that. I'm not sure what that, oh, that's the hand in the glove model. There we go. So this is the hand in the glove model where the hand fits into the glove and then the glove changes its shape to mold to the hand. And the enzyme, when it binds to the substrate binds on the active site, the enzyme changes its shape. And that happens in molecules, that when the enzyme binds to the substrate, it changes the enzyme shape, three-dimensional shape. And there you can see the enzyme changing. For those of you who are really close, you can notice that this molecule looks a little different than that molecule. This molecule is flatter than that molecule. And this I call the shapeshifter's hand in the glove model of how an enzyme functions. And that is, yes, the glove is molding to fit the shapeshifter's hand, but the shapeshifter's hand is also molding to fit the glove. Those of you who know Star Trek Deep Space Nine know what the heck I'm talking about when I'm talking about a shapeshifter. It was a creature that could change its shape. And so the hand changes when the hand goes in the glove, or what I'm exactly saying is the substrate changes its shape when the substrate binds to the enzyme. And it's true the enzyme also changes its shape. That's why I said it's the shapeshifter's hand in the glove model. Any question about any of that? All right. There are different factors that can influence the amount of activity an enzyme has. An enzyme has to be at the correct temperature, the correct pH, the correct salinity, has to have the correct heavy metals around at low concentration. And if heavy metals are around at higher concentration, or heavy metals that are toxic are around, then the enzyme will have less activity. The enzyme will have most activity when there's no alcohol around, there's no UV radiation, and when heavy metals or alcohol or UV radiation are around, or even the incorrect temperature, the incorrect pH, and the incorrect salinity, then the protein of the enzyme can start unwinding and taking on a different three-dimensional pattern. And this we call protein denaturation, where the protein is denaturing. The enzyme will also have its best enzyme activity when the substrate concentration is high, or the enzyme concentration is high, and when poisons are not around. If poisons are around, like cyanide or fluoride, they can inactivate the enzyme. So I guess what I'm trying to say is the enzymes in the substrate do not work in a vacuum. To be able to work at all, you have to be at the right temperature, the right correct pH, the correct salinity. And if you're not at these, the enzyme may function, but it won't function at its optimal rate. And that is if it functions at all. Like if you took our enzymes and subjected them to boiling water, our enzymes would not work at all. The enzyme would be totally denatured. Any questions about the factors influencing enzyme activity? All right, you can have inhibition of an enzyme. That inhibition can be competitive inhibition or non-competitive inhibition. Non-competitive inhibition is also called allosteric inhibition. And then lastly, we'll talk about feedback inhibition. So there we have an active functional protein, which is an enzyme taking on a three-dimensional shape. This enzyme will work at its optimal rate, meaning its best rate. And then that enzyme can be denatured, taking on another three-dimensional shape. You can have the enzyme denaturing. Excuse me again. Blow my nose again. See if that helps. You can have the enzyme denaturing by having it at the incorrect temperature or the incorrect pH. This enzyme has its optimal rate at around 37 degrees. And when you change the temperature a little bit, the enzyme stops working at its optimal rate and works at a lower rate. You change the temperature further, the enzyme starts denaturing further, meaning it's not totally denatured. This part is only slightly denatured. More denatured here, where the enzyme is working at an even lower rate than this rate here, which is lower than the optimal rate. And then you can go down to change the temperature dramatically, such as below 10 degrees Celsius or above 50 degrees Celsius. And then the enzyme will be totally denatured, where it cannot function. Any question about any of that? For this one here, this will happen if you heat up the enzyme greater than 50 degrees Celsius, because the extra energy in this will break the hydrogen bonds forming, will break ionic bonds, will break other forms of attraction like van der Waals forces. You can totally denature the enzyme, and that usually happens like when you boil our enzymes. You can also put the enzyme in low temperature, and the enzyme will totally denature. At low temperature, probably what's happening is that the hydrogen bonds are forming in the molecule where they should not be. Ionic bonds are forming in the molecule where they should not be, in which case the enzyme may not be in this type of a three-dimensional shape where there's almost no force of attraction between them. When you put the enzyme in a lower temperature, you will actually get the three-dimensional shape changing, where hydrogen bonds will be forming which should not be there. And what happens is, so let's make it the three-dimensional shape. Can you guys see that? Anyone? Can you see what I'm trying to show you?
Yes.
Okay. What happens when you put it in the lower temperature, you're getting other bonds forming, and the three-dimensional shape will change. And generally, if you lower the temperature, you'll have other forces of attraction which should not be there happening.
I'm having a hard time seeing what you're showing because it's sometimes covered by your name.
Oh, well, sorry about that. Let me stop the share and then I can move it. You see my name there? I don't see my name. All right. So, gotta go it over my face so I can't see it. So here's the enzyme in the optimal temperature or the optimal salinity. It's in a three-dimensional shape. I'm trying to make it, I don't know, kind of roundish, which I can't show too well with my hand. There we go. And then you lower the temperature. It can be even more compact, changing its three-dimensional shape, where our hydrogen bonds will be forming that should not be there. Ionic bonds may form that should not be there. And then you can also raise it, in which case you get the protein having, see, its normal shape was something like this. Ah, come on. Guess you can sort of see it. And it'll be in a different shape, and the protein will just unravel. There will be almost no forces of attraction in the protein. Any question about any of that? Oops, wrong one. Anyways, with different temperature, this enzyme has a optimal temperature around 37 degrees. So this could be one of our enzymes because they have an optimal temperature. Most of our enzymes have an optimal temperature at 37 degrees. And then when you move away from it, you'll get less activity. The enzyme will be starting to denature. The further away you move from optimum, the less enzyme activity, the more denatured the protein, until you're fully denatured, in this case above 50 degrees or below 10 degrees. The same thing happens with pH. Here we have an enzyme that has an optimal pH around pH 5. And when you move away from that pH,
Did you go back to screen sharing?
I'm sorry, I thought I was sharing. When you go away from the optimal pH, you get the enzyme starting to denature. The enzyme will have less activity. You go further in the pH, you'll have less enzyme activity, more denaturation of the enzyme, until you go to extreme pH, where the enzyme will be totally denatured. You'll have no enzymatic activity. Here's a question for you. Where do we have an enzyme which works at an optimal pH of pH 5? I'll give you a hint. The stomach is much lower than pH 5. Anyone have a guess?
The mouth?
Yeah, that would be a good guess. Yeah, your mouth will be acidic if you have bacteria in the mouth making acids, and that normally is the case. I was thinking more of the skin, but the mouth is a good example too. And we do have enzymes on our skin which can work best around pH 5 because that's the temperature or the pH of our skin. In our mouth, I don't know of an enzyme which works best around pH 5, but we might have one or two. I don't know. Any question about any of that?
I, the screen you're sharing is just black, or is
Just
it just
black?
Yeah.
You saying something?
I can see the screen.
I
Oh,
can too.
okay.
Um,
So it might just be mine. Sorry.
you might need to restart your Zoom.
Okay.
I'm not sure what the problem is because I'm sharing my screen and other students are seeing it.
Perfect. Thank you.
Okay. All right. So there are factors that influence the amount of enzyme activity. Substrate concentration is one. When we have very low substrate, the enzyme activity will be very low. And that's because the enzyme, the chance of it meeting the substrate is pretty small. And so there is not much activity of the enzyme. And we can increase that activity of the enzyme by increasing the substrate concentration. As we increase the substrate concentration, the amount of enzyme will increase. The amount of enzyme activity will increase. That will happen up to a point, but as we increase the substrate concentration, the amount of enzyme activity starts to slow until we get to about right here, where when we increase the substrate concentration, the amount of enzyme activity does not increase at all. The reason why that happens is because at this point, all of the enzyme is already bound to the substrate. And so if all of the enzyme is already bound, once you increase the substrate concentration, you do not increase the enzyme activity at all because all of the enzyme is already bound to the substrate. So it does not increase the activity at all. Any question about any of that? All right, let's talk about competitive inhibition or inhibition and how it influences the amount of enzyme activity. Competitive inhibitor is an inhibitor that competes with the substrate for the active site of the enzyme. We actually have some drugs which compete with the active site with the substrate for the active site of the enzyme. And then the drug will bind to the enzyme, not allowing the substrate to bind to the enzyme. And that is competitive inhibitor. The inhibitor is competing with the substrate for the active site. Any question about competitive inhibition? All right, let's talk about non-competitive inhibition, also called allosteric inhibitor. This is where the inhibitor binds to the enzyme at a site other than the active site. So the non-competitive inhibitor binds here, called the allosteric site. That changes the three-dimensional folding of the enzyme, changing the three-dimensional structure of the active site. And now the active site cannot bind to the substrate. And that's how the allosteric inhibitor inactivates this enzyme, changing the folding pattern of the enzyme so the enzyme cannot bind to the substrate. Any questions about competitive or non-competitive inhibition? If you have an inhibitor present, obviously the enzyme will not work at its functional optimal level. It will work at decreased level. And if you have enough of the inhibitor, the enzyme will not function at all, will have no activity. let's talk about feedback inhibition. This is a type of non-competitive allosteric inhibition. Guess I can leave that up. Oops, that's way too big. Come on. Ah, come on. I'm going to go very slowly, but we're going to do it that way because there we go. Yep, let's blow it up because I need to show you it very clearly. So we have a substrate and it binds to enzyme 1. Enzyme 1 converts the substrate to intermediate molecule A. Another enzyme, enzyme 2, converts intermediate A to intermediate B. And then we have a third enzyme which binds to intermediate B and makes the end product. The end product in feedback inhibition can then bind to enzyme 1, changing the folding pattern of enzyme 1 so that enzyme 1 no longer binds to the substrate. If you have feedback inhibition going on in a cell and there's little end product, all of the enzyme will be active, or at least most of it, and then we'll make more end product. As that end product builds up in the cell, it'll start binding to enzyme 1, shutting down enzyme 1, and then we'll make less end product. So our cells have feedback inhibition in them to keep the amount of end product within a certain range. When the range is too low, we'll make more of the end product because more of the enzyme 1 will be active. And then as end product 1 builds up in concentration, we'll start shutting down more of enzyme 1 because the end product will bind to enzyme 1, shutting it down, and then we'll make less end product. And the cells have this feedback inhibition in them to keep the end product at a certain concentration within the cell. Once the end product drops below a certain concentration, we will have more of the end product made, and as more of the end product gets accumulating in the cell, we'll start shutting down the chemical pathway to make the end product. And then eventually, when we have enough end product, all of enzyme 1 will be shut down, and we won't make more end product. And that's the way feedback inhibition works. Any question about any of that? All right, let me state that in cells, feedback inhibition is usually seen this way, where the end product itself binds to the first enzyme, shutting down the enzyme. Why? Because if you had the end product binding to enzyme 2, you will make intermediate A, and that will just waste the cell's energy. So we have the end product binding to enzyme 1 because this is the most energy efficient pathway. And then if you had another molecule besides the end product binding to enzyme 1, you wouldn't really regulate the amount of end product that's being made. So you always need the end product to be the molecule binding to enzyme 1 to shut it down. And that's feedback inhibition. You have a lot of feedback inhibition going on in your cells. Any question about any of that? All right, let's change the topic a little bit and talk a little bit about ribozymes. We've talked about protein enzymes. Now let me state that there are some enzymes which are not protein. Most enzymes are made up of protein, but a few enzymes are made up of RNA, and these we call ribozymes. They are an RNA enzyme. This RNA cuts and splices RNA or another molecule, and the ribozyme functions as an enzyme, and that the RNA molecule, being the ribozyme, actually has an active site that binds to a substrate. And then the ribozymes are not used up by the chemical reaction. And so once the ribozyme converts the substrate to the product falls off, and then that ribozyme can bind to another substrate, the same as a protein enzyme. Any question about any of that? All right. Datura does not mention it. If you don't know, that's your textbook author. But there is one RNA ribozyme that we will actually talk about in this class. And that is when a protein is being made and the ribosome actually will bind the amino acids together to form the growing polypeptide, and then we'll take that growing polypeptide off the tRNA and then bind it to the next amino acid coming on a tRNA. So we'll talk about that enzyme, and that enzyme is actually a ribozyme. It's the RNA portion of the ribosome that is doing the enzymatic activity, making the growing polypeptide that will actually form the finished protein. Any questions about any of that? All right, let's change the topics again and let's talk about a little bit about metabolic diversity. There is metabolic diversity among organisms. We're talking about all organisms, but obviously we're going to specifically talk about microbes. All organisms, including microbes, require the following sources. They require an energy source and they require a carbon source. Based on their energy source, organisms can be classified on where they get their energy from. We have phototrophs, and these organisms derive their energy from light. And then we have chemotrophs. These organisms derive their energy from either an inorganic or an organic chemical compound. If you don't know, we are a chemotroph. We derive our energy from an organic chemical compound, such as glucose. Green plants, everybody knows, derive their energy needs from sunlight, and so green plants are a phototroph. We also have green bacteria, which are phototrophs. Any question about any of that? All right. And then we have some chemosynthetic bacteria that derive their energy needs from an inorganic chemical compound like H2S, a hydrogen sulfide gas, something that's toxic to us, or maybe from sulfate or ammonia or iron. The point is there are some chemosynthetic bacteria that derive their energy needs from an inorganic chemical compound. And that's a little bit different than us or another chemotroph that obtains their energy needs from an organic chemical compound like glucose. We can also classify an organism based on where it gets its carbon source from. Organisms can be classified as an autotroph if they get their carbon from CO2, either dissolved in the air or dissolved in the water. Like green plants get their carbon from CO2, and so green plants are an autotroph. And then there are organisms that get their carbon source from an organic chemical compound, such as glucose. And we are heterotrophs. We get our carbon from an organic chemical compound such as glucose. Any question about any of that? All right. If ever you get confused, simply split the molecule down into where it gets an energy source, a phototroph or a chemotroph, and then where the organism gets its carbon source, either from CO2, from the air, an autotroph, or an organic carbon source being a heterotroph. Any question about any of that? All right, you can then get metabolic diversity among organisms by considering both where they get their energy and their carbon source from. And then you get the organism defined as a photoautotroph, a photoheterotroph, a chemoautotroph, or a chemoheterotroph. Any question about any of that? All right, the photoautotrophs are one that's easy to explain. Most people understand this because they know plants. They are a phototroph because they get their energy source from light, and they are an autotroph because they get their carbon from carbon dioxide. And green plants and green bacteria are photoautotrophs. And then we have the next easiest one, a chemoheterotroph. They get their energy source from a chemical compound, either an organic chemical compound such as glucose or an inorganic chemical compound such as hydrogen sulfide, iron, sulfate, a nitrogen compound. And then the chemoheterotrophs get their carbon from an organic chemical compound such as glucose. We are a chemoheterotroph. Why this is probably easy to understand. We get our energy needs from glucose, and we get our carbon also from glucose. The chemoheterotrophs, many of them, have the same molecule for both their energy needs and their carbon source. And that's often the case for a chemoheterotroph. So the fermentative bacteria, animals, protozoa, fungi, and bacteria like E. coli are chemoheterotrophs. But there are some chemosynthetic bacteria which are chemoheterotrophs, and they get their energy needs from an inorganic chemical compound, such as hydrogen sulfide gas, sulfate, nitrate, or iron. And they get their carbon from an organic chemical compound such as glucose. Any question about any of that? All right. And then we have the chemoautotrophs. They're the next most easy to explain. These are some of the chemosynthetic bacteria, and they get their energy source from an inorganic chemical compound, such as hydrogen sulfide gas, sulfate, or iron, and they get their carbon from CO2. And so the chemosynthetic bacteria are split in the chemoautotrophs and the chemoheterotrophs. If they get their carbon from CO2, they are a chemoautotroph. If the chemosynthetic bacteria gets their carbon from an organic chemical compound such as glucose, then they are a chemosynthetic bacteria, but they are a chemoheterotroph. Any question about any of that? All right, I stated a chemoheterotroph that's easy to remember. People are chemoheterotrophs. Let's state a chemosynthetic bacteria, which is a chemoautotroph that all of you have seen. The iron oxidizing bacteria in the toilet tank, not the toilet bowl, but when you lift up the lid of the toilet tank, storing the water that we flush down. You see that brown or yellowish scum on the toilet tank? That is iron oxidizing bacteria. That is a chemosynthetic bacteria. That is a chemoautotroph. It uses iron to get its energy needs, and it gets its carbon from CO2 dissolved in the water. You've all seen that, right? Anyone not seen that? Not hearing anything, so I'm going to assume everyone's seen that. And that is a chemoautotroph, that brownish or yellowish scum on the toilet tank. The photoheterotrophs are the most difficult to explain because you probably never come across a photoheterotroph before. They are a phototroph, so they get their energy needs from light, and they are a heterotroph, so they get their carbon from an organic chemical compound such as glucose. You've probably never heard of these, but the green and purple non-sulfur bacteria are photoheterotrophs. Any question about any of that? All right, let's pull this slide up. So we have all organisms. We can divide them by where they get their energy source. We have phototrophs. They get their energy needs from light. We have chemotrophs. They get their energy needs from a chemical compound, either an organic chemical compound such as glucose or a inorganic chemical compound. If they get their energy needs from an inorganic chemical compound, we call them a chemosynthetic bacteria. You can then separate the phototrophs by where they get their carbon source. If they get their carbon source from CO2, they are a photoautotroph. You guys all know the photoautotrophs because green plants are photoautotrophs. If, on the other hand, they get their carbon from an organic chemical compound such as glucose, they are a photoheterotroph. You guys don't know any photoheterotrophs, but I'm telling you the green non-sulfur bacteria are photoheterotrophs. You can further split
I had a
the...
question.
Yeah,
Were we ending the lecture at the normal time today?
6:50, yeah.
Okay.
That time? I didn't think it
Yeah.
was. Oh yeah, it just turned 6:50. All right, I'll stop here and say you only need to know the portion in red, so you can divide the chemotrophs where they get a carbon source. If they get a carbon from an organic chemical compound, they're a chemoheterotroph. If they get their carbon from CO2, then they are a chemoautotroph. All right, any questions? If not, I'll stop here and we'll continue with this slide in our next lecture. I'll see you. We'll start at the lab a minute late because I'm ending this one a minute late. Any question about that? All right, I'll see you.